And today let's learn something about Chapter 4 Learning, Memory, and Emotions. And before we start, we should learn something about a patient. And the patient, known for most of 5 decades only by his initials, HM, led to one of the most significant turning points in 20th century brain science. The understanding that complex functions such as learning and memory are tied to distinct biological processes and regions of the brain. Following a childhood blow to the head, Henry Molaison developed severe seizures. In 18 years later, still experiencing debilitating symptoms, he underwent an experimental procedure that removed sections of his medial temporal lobes, including most of his two hippocampus, and the seizures abated. But Melason was left with permanent amnesia. He could remember scenes from his childhood, some facts about his parents, and historical events that occurred before his surgery, but was unable to form new conscious memories. For example, if Melissa met someone who then left the room, within minutes he had no recollection of the person or their meeting. He experienced every aspect of his daily life, eating a meal, taking a walk as a first, yet his intellect, personality, and perception were intact, and he was able to acquire new motor skills. Over time, he became more proficient at tasks such as tracing patterns while watching his hand movements in a mirror, despite the fact that he could never recall performing the task before. Studied by neuroscientists for 50 years until his death in 2008 at age 82. Melissa's intact abilities as well as his impairments provided evidence for the roles of the hippocampus and hippocampal region in converting memories from short-term to long-term, paving the way for further exploration of brain networks encoding conscious and unconscious memories. And about learning and memory, our understanding of how humans learn and remember is far from complete, but researchers are uncovering intriguing new details about the mechanisms, limits, and architecture of memory information. Thanks in part to HM, scientists now know that the medial temporal lobe, which includes the hippocampus and hippocampal regions, works with other regions of the cerebral cortex, the brain's outermost layer, to form, organize, consolidate, and retrieve memories. The four major lobes of the cerebral cortex, frontal, parietal, temporal, and occipital, process sensory information such as smell, taste, sight, and sound. Associative regions in the cortex integrate these sensory inputs, enabling us to understand our environment and encode memories. And first, memory. About the long-term memory, it's divided into declarative memory and non-declarative memory. And the declarative memory is memory for facts, data, and events. Such conscious memories are called declarative memories because you can consciously recall and describe the information. Declarative memories can be semantic or episodic. Semantic memories consist of the cultural knowledge, ideas, and concepts you accumulate about the world. For example, names of state capitals, word definitions, how to add and subtract, or dates of historical events and their meaning. This type of memory involves cortical regions well beyond the hippocampus. Episode memories are unique representations of your personal experience. For example, mentally recalling the sights, sounds, time, space, and emotions associated with an experience involves episodic memory. And in this picture, there are several different kinds of memory, and they are processed by different areas of the brain. The hippocampus, hippocampal region, and areas of the cerebral cortex work in tandem to produce memories of facts and events. Other kinds of memories, such as emotional and behavioral memories, are handled by other parts of the brain, including the amygdala, striatum, and cerebellum. And then non-declarative memory, also known as implicit or procedural memory, is stored and retrieved without conscious effort. You use this type of memory when you perform learned motor skills like speaking or reading a book. HM did not lose this type of memory, as evident in his ability to acquire new motor skills, even though he couldn't remember doing them before. The fact that HM and other people with amnesia show deficits in some types of memory but not others indicates that different types of memories are encoded in separate but interacting regions of the brain. Motor skill learning, for example, involves many areas of the brain, but three are especially important: the basal ganglia, the habit center of the brain, the prefrontal cortex, and the cerebellum, an area at the back of the brain involved in motor control and coordination. And your brain is able to form memories and rewire itself in response to experience because circuits in your brain change at synapses, the tiny gaps across which neurons communicate with chemical and electrical signals. The ability of synapses to remodel themselves is called synaptic plasticity. And encoding a new long-term memory involves persistent changes in the number and shape of synapses, as well as the amount of neurotransmitter released and the number of receptors on the postsynaptic membrane. And second, about the emotions. In emotional memory, considered another type of non-declarative memory, learned emotional responses become attached to stimuli over time after repeated exposure. In the 1970s, anthropologist Paul Ekman identified what he called the six basic emotions: anger, fear, surprise, disgust, joy, and sadness. While scientists have since disputed the exact number and attributes of human emotions, whether emotions are consistent across cultures, or even how to define an emotion. Their research has linked some neural circuits to physiological responses that help us survive, interact, set goals, and initiate actions. And human actions are driven by necessities: food, sleep, sex, avoidance of pain, and by rewards. But our responses and actions are not always logical. While little is known about exactly how the brain transforms feelings into decisions, researchers have developed theoretical models about decision making. Effective decision making involves choices under risky and uncertain conditions. An active area of neuroscience research is investigating how the brain balances reward and risk, and how emotional state affects this balance. Emotionally centered decision making changes with age, possibly because the lateral prefrontal cortex, responsible for self regulation, matures gradually in adolescence. Teens developing brains and high sensitivity to peer acceptance may be related to their increased tolerance, tolerance for risky behaviors. Other, other adults may also make more risky decisions as PFC function diminishes with age. So one of the motivation of emotions are affective decision making, and the second motivation, dopamine and reward pathways. Although relatively few neurons in the mammalian central nerve system generate the neurotransmitter dopamine, this dopaminergic neurons influence multiple brain functions, including voluntary movement and a variety of behavioral processes such as mood, reward, addiction, stress, and memory. When something is very rewarding, we are more likely to remember it. That is because dopamine influences the synapses in the entire reward pathway, the hippocampus, amygdala, and the prefrontal cortex, to create emotional associations with rewards. And the, and the mesolimbic pathway, sometimes called the reward pathway, is a major pathway for dopamine, connecting the midbrain's ventral tegmental area, also called VTA, to the nucleus accumbens. It is involved in cognitive processing of reward and motivation. Neurons that release dopamine are activated in response to signals that a reward will be given. Surprisingly, it's not the reward itself, but the expectations of a reward that most powerfully influences the emotional reaction. Reward learning occurs in response to something unexpected, when the actual reward differs from what was predicted. If a reward is greater than anticipated, dopamine signaling increases. If a reward is less than expected, dopamine signaling decreases. In contrast, a correctly predicted reward does not elicit changes in dopamine signaling, and all remains the same. And interestingly, recent research shows that dopa, dopaminergic responses vary among people. Some people's brain respond more strongly to rewards than punishments, while others respond more strongly to punishments. The, the amygdala has been impl- implicated in various aspects of reward learning and motivation. Researchers at Vanderbilt University found that go-getters who are more willing to work hard have greater dopamine signaling in the striatum and prefrontal cortex, two areas known to impact motivation and reward. While the brain's reward system typically reinforces behaviors associated with rewards and prevents behavioral leading to punishment. Aberrant circuitry can lead to inappropriate aggression, a symptom of some neuropsychiatric disorders. For example, the lateral habenula, a major node in the reward circuitry, appears to encode punishment by inhibiting dopamine release, and dysfunction of the lateral habenula has been linked to disorders involving inappropriate aggression. The amygdala has also been associated with negative emotions. Stimulating some areas can trigger rage and aggression, while removing specific sections of the amygdala will make lab animals more docile. Recent studies in lab animals have also suggested that aggression can result from inappropriate activation of the brain's reward system in response to violent social stimuli. And this is a picture that shows the brain's reward system.